Protein synthesis. Transcription is the first half of the process of protein synthesis. It involves the copying of a DNA strand into a molecule of mRNA or messenger RNA. Transcription occurs within the nucleus in a eukaryotic cell. There are two steps. First, DNA becomes pre-mRNA. Next, the pre-mRNA undergoes modifications to become mRNA and leave the nucleus. Pre-mRNA. To create pre-mRNA, most of the work is done by one enzyme. While in DNA replication you need several enzymes like ligase, DNA polymerase 1 and 3, helicase, and RNA primer, in transcription, almost all of the work is done by RNA polymerase alone. It creates the nucleotide sequence that will become mRNA. To do this, RNA polymerase must first find the region on a DNA strand called the promoter. Promoters are specifically associated with the type of gene they will code, so when RNA polymerase needs to find a certain gene, it can always start at the promoter region for that gene. RNA polymerase will attach to the DNA strand once it has found the promoter region. From this point, it begins to separate the strands of DNA and code for the new pre-mRNA strand from 5' to 3' at the same time. It continues to do this until it reaches a terminator region on the strand. In this region, the RNA polymerase lets go of the newly created pre-mRNA strand and it is sent off for modifications. Modifications. So, I mentioned earlier that the pre-mRNA must undergo modifications to become mRNA and leave the nucleus. What are these modifications and how are they formed? There are two main modifications, the 5' cap and the poly-A tail. The 5' cap is a modified guanine base at the starting end of the pre-mRNA strand. This cap is made to ensure that ribosomes can attach to the strand for translation. The poly-A tail is a series of multiple adenines added at the end of the strand. This makes it less likely that the strand will become damaged. Another modification that can happen involves the splicing of introns. Introns are junk DNA, or DNA that doesn't express any genes. In RNA modification, they're spliced out so that only exons, or genes that do express traits, remain. Once this process is done, the mature mRNA leaves the nucleus and is ready for translation.
Translation is the second half of protein synthesis, and this is the process of translating mature mRNA to a sequence of amino acids, which eventually codes for a specific protein. This occurs right after transcription in eukaryotes, and it takes place in the cytoplasm. There are three stages of translation: initiation, elongation, and termination. Before we get into the steps of translation, it's important to know what's involved. The main components are mRNA, ribosomes, tRNA, and amino acid tRNA synthase. mRNA is kind of like a blueprint. It has all of the necessary information for the tRNA, which is more of a builder, to be able to transfer amino acids. Ribosomes, made up of proteins and rRNA, are the structures where proteins are built. mRNA nucleotides are read in groups of 3. Each group forms a codon. Each codon eventually codes for a specific amino acid. One really important example of a codon is AUG. This specifies the amino acid methionine, which acts as a start codon, which is what starts the process of protein construction. There are a few exceptions. Some codons don't code for amino acids at all, and they tell a cell when a polypeptide is complete. These are known as stop codons. Finally, tRNA molecules have anticodons. These go along with codons from mRNA. And amino acid tRNA synthase links tRNA molecules with their corresponding amino acids. Now that we know the major components of translation, let's put them into context. First, the small subunit of a ribosome binds to the 5' cap of mRNA so that it can scan for the start codon. Once this is recognized, the large subunit binds at two sites, the P site, which holds the peptide chain, and the A site, which accepts tRNA. In elongation, mRNA is read one codon at a time, and each codon's corresponding amino acid is added to a growing polypeptide chain. This involves a continuous cycle, powered by GTP, of incoming and outgoing tRNA, which initially binds at the A site and translocates to the P site before finally leaving the ribosome. As new tRNA approaches, the enzyme peptidyltransferase forms peptide bonds to attach it to the start codon. As tRNA cycles through elongation, eventually a stop codon reaches the A site. No more molecules bind to these, so the tRNA in the P site goes through hydrolysis as the polypeptide is released into the cytoplasm, and the ribosome separates, ready to perform translation again. The polypeptide sometimes needs to go through modification, which can consist of folding into a 3D structure, processing, which removes amino acids, getting shipped to the right place in the cell, or combining with other polypeptides. And all of this has to happen before it can act as a functional protein. And that's it. Protein synthesis is a complicated process, but it's a universal process that happens really quickly, and just one small change can make a really big difference.